studies as to the factors responsible for the disease variability in ALGS.
Yeah, thank you, Roberta, for giving me this opportunity to present work that's sort of been ongoing and sort of we've been limping along on this project for quite a while now. But I'm excited to kind of share what we know. And really, to be frank with you, Roberta asked me about 2 weeks ago to fill in a slot that opened up, so I'm kind of a backup. So if I don't sound as good as the other speakers, it's because of that. Yeah. So I'm really going to share with you my sort of thoughts on why there's so much variability, and I think that's been on a lot of people's minds. So it's not just me who's thinking about that. And the only thing that I can really share with you that might be unique is that I've been studying mouse kidneys and mouse kidney disease related to Alagille or related to Notch signaling mutations. And so there's stuff there that we've observed, and that's kind of really what I'm going to be sharing with you. I don't know how much it translates to the human setting, so just that's my disclosure. I don't see patients, I study mice. Is it the other one? Or? Okay, so yeah, I don't have any conflicts of interest. So as we've heard many people talk about Alagille syndrome, Dr. Alagille kind of tried to help us by simplifying the disease to a certain extent. So there are a lot more symptoms, and actually one of the things that he left out is the kidney disease, right? So I would say he threw out the baby with the bathwater in this, but to a certain extent. Obviously the liver is an important thing, and that's on here. But so some of the studies that so it's actually, even if you go back to some of the early studies, there is instances of polycystic kidney disease reported in some of the very first instances of Alagille syndrome. But it was really Nancy's Dr. Spinner's work that identified that it's not signaling pathway genes, mostly Jagged Wan, and Notch2 that are involved in this disease. And what that really opened up was the genetic basis kind of revealed that there's other diseases that are also important and associated with Alagille syndrome, one of them being the kidney, the other being the vascular defects. So really today I'm going to focus on the variability in the kidney disease, and one of the important pieces of work done by Dr. Kamath was to sort of do a retrospective study of patients who have Jagged Wan mutations. And what she identified, and I've heard this question, is only Notch2 related with kidney disease, and that's not really true. So this is one of the evidences that Jagged Wan is also related to kidney problems. So about 40% of people with Jagged Wan have some sort of a kidney issue. The actual kidney issue is kind of variable, and one of the most common ones is the small abnormal or dysplastic kidneys with or without cysts, and that occurs in 60% of ALGS patients. So if you put those two numbers together, 40 and 60, so you would expect about a quarter of Jagged Wan patients to have small cystic kidneys. The other kind of issues, you see renal tubular acidosis, hydronephrosis. One of the things that we see in our mice is this urine concentrating defect, and this is something that I've been wondering for a long time whether it actually happens in humans or not. There is this reference to a South American paper that there are defects in urine concentration. This is something that I'm kind of really interested in, so if you think you might be having such issues, I'd really like to know. This is one of the things that the CORDS registry is kind of recording that, and I've already seen that there is at least one patient who's reporting this kind of a thing. So this is one of the reasons for having these registries. So it's not just Jagged Wan. The Notch2 has also been associated with kidney diseases. And the very first paper from Dr. Nancy Spinner's lab actually sort of highlighted the kidney disease with the Notch2 mutation. So these are some of the Notch2 mutations that you see. And initially, so these are all patients who are clinically diagnosed with Alagille syndrome, and they have various Notch mutations, Notch2 mutations. Not all of them have kidney disease. In this family, you see that there are two different family members that have the kidney disease. One actually has sort of an adult kidney disease, and the other one is congenital, or the child has the severe cystic kidney disease. So even with the same mutation, there's somewhat of a variability. Here's another family with again, there are three members. One is unaffected in terms of the kidney disease. The other one has adult renal failure, and then there's a third. As a child, they have sort of severe kidney disease. So again, there's high amount of variability, even with the same mutation, and there's also variability in terms of whether the disease occurs or not. More recently, there have been studies using whole exome sequencing. So CACUT is not Alagille syndrome, but what it stands for is congenital anomalies of the kidney and urinary tract. So basically, by ultrasound, they're finding that newborns are having abnormal kidneys, and when you sequence their exomes, they actually found point mutations in the Notch2 gene. So these patients are not thought to have Alagille syndrome, but they have the kidney issues. So really, there are three ideas that I want to kind of talk about, and I put lifestyle and diet up here, and this is the thing that I know the least about, actually, and I'll try to discuss it towards the end. But there are some mouse studies that we're starting to do that sort of suggest that the type of diet actually influences Notch signaling possibly, which is important for the adaptability or the adaptive capacity of the kidney, okay? And that might, it's sort of at the beginning stages, those studies, but it's sort of suggestive that certain diets may be an issue for Alagille syndrome patients, and that's taking it to the extreme. We don't know that for sure. But that's where I'm getting at, with lifestyle being one of the possibilities for why there's so much variability, right? The other, of course, we've heard about the genetic variations, and the way that I'm thinking about this is, yes, so people have mutations in Jagged Wan or Notch2, but do we have additional variations, much like Dr. Spinner was talking about the differences in severity in liver disease? I'm kind of interested in the kidney disease. Is that one thing is that we might have mutations in genes that mediate organ-specific functions of Notch signaling, and so you might have a more severe kidney or liver disease because of that gene being specifically functional in that organ. Or it could be that the severity is across the board. The mutation may be in one of the core Notch signaling pathways, in which case the severity is likely to increase in multiple tissues. So this third concept is kind of a hard one to kind of talk about, and it's not really easy to understand because it sort of deals with statistics to a certain extent, and it also sort of deals with the idea of sort of systems biology and kind of thinking about systems and how robust they are, and how they can sometimes become random. So what I'm trying to suggest here is that the kind of mutations that we have so far associated with Alagille syndrome in Jagged Wan and Notch2, they're not complete null mutations. As you heard, they're sort of dominant where one allele is being inactivated. So you're dialing down the level of Notch. And so this is actually sensitizing the level of Notch signaling. And what that does, at least in the mouse studies, what we see is that allows for a lot of, that allows for chance and a lot of other normally minor factors to play a bigger role in the actual outcome, okay? So it's sort of a hard concept, and I'll try to come back throughout this presentation to this idea. So the sensitized background, it kind of allows for this randomness, and that's kind of why there is variability, all right? But these three factors are not sort of exclusive, so all three could be happening. And in fact, when you're in a sensitized background, it's more likely that there are genetic variants that normally don't matter that start mattering. And the same with the diet. Diets may not normally matter, but once you're in the sensitized background, it might actually matter. So in terms of the clues, so one big clue is that we have is that the degree and the timing of Notch signaling activation within the developing kidney kind of predicts the severity of the kidney disease. Okay, so what do I mean by this? I'll kind of show you. So we can actually control when and where we're inactivating Notch, and we're only inactivating it in the developing mouse kidney, okay? So, but before I tell you that, I mean, most of you guys have seen this, but I kind of need to emphasize this because you have to understand that basic research is becoming fast, it's almost becoming a dinosaur-like thing because there's hardly any funding for basic research. And I kind of have to emphasize with you guys that this is something that we all need to be supportive of because there's a wealth of knowledge that has come from basic research, such as from the fruit fly genetic studies. As you've heard from Dr. Spinner, she's done an amazing job of linking Alagille syndrome to the jagged and the notch signaling. But the next step really is it opens up a whole set of knowledge about the notch signaling pathway, and that really comes from fruit fly studies, where they actually identified the different components of the signaling pathway. And so you kind of have to give credit to the Drosophila geneticists who, as early as 1914, identified this notched wing, and people have cloned that and identified what that gene codes for. And by genetics, people identified the different components of this signaling pathway, which is evolutionarily conserved in humans and mice. And so these model organism studies, you might think, okay, why the hell are we still wasting our tax dollars studying fruit flies? It's because it provides a wealth of knowledge. All right, and in fact, I like to kind of joke around that really the first Alagille-related symptom is this notched wing, okay? Because, so if you might think that, okay, you're a human being, kind of, you're dreaming that you're a human, but you might be a fruit fly, right? And kind of escape into that. But if you have a notch mutation, you might still not be in a perfect shape in that you might have that notched wing. So even in the fly, if you lose both copies of notch, and only in females can you lose both copies because this is actually on the X chromosome. So the males actually with a mutation are not going to survive. It's only the females that have that notched wing. So if both copies are gone, the embryo isn't going to survive. And it's the same in the human setting. So this is a slide that I stole from Rafi Kopan about 10 years ago. But it really is to point out how the signaling pathway works. So it's a way for cells to communicate between each other. And as you heard from Dr. Spinner, we all started from a single cell, and now we're multicellular. And this is one of the pathways that sort of tells cells to become different from each other. It tells cells that are next to each other, okay, you become a hair cell, I'm going to become the neuron. So it's sort of a way of distributing the functions for the cells. So you can think that that's actually a very critical signaling pathway, and that's kind of why we have so many different issues going on when you have that mutation. So here's one cell with a ligand sticking through the membrane. So Jagged Wan is actually a protein that's sticking through the cell membrane. And the Notch 1, 2, 3, and 4 are also a transmembrane protein sticking through the adjacent cell membrane, all right? So what happens is the ligand binds the receptor, and it literally pulls on the receptor, and it actually endocytoses. And that opens up a negative regulatory region here for this yellow enzyme called atom metalloprotease to cleave it, okay? And that creates a stub-like protein, which then is a target for gamma secretase, which further chews on it and releases the intracellular domain of Notch. And Notch can then go into the nucleus. So here's the nucleus, and there it actually competes for binding to RBPJ, this red protein on DNA. And when it binds to RBPJ on the DNA, it can turn on genes, all right? And it turns on different genes depending on when and where it is actually functioning. So it seems like a simple pathway with the ligand, receptor, and these few different components like the DNA binding factor, but the genes that actually get turned on, there's a variety of them depending on when and where the signaling is happening, and that's sort of the complicated aspect of the pathway. So one of the things, I don't really want to get into kidney development, but this is sort of showing you how nephrons are going to form. And we have genetic tools to kind of inactivate Notch at different time points, and that's all I want you to get out of this slide. So these are tools in which we can target only these blue cells that are going to form the nephrons. We can either target it very early, or we can sort of target it in a delayed manner. using the 6-2 Cree, right? And we even have a way of targeting adult nephrons using this mouse line. So what happens if you, normally this is a nephron, okay, it has different segments, and this is kind of a stain looking at the proximal tubules in a wild type. If you get rid of RBPJ or Notch2, if you inactivate it very early, you actually don't get the proximal tubules, so this green stain is no longer there. So you actually form non-functional nephrons, and these mice don't survive past 2 days because they don't have functional kidneys. If you delay the inactivation, now you're getting rid of either Notch2 or Notch1 using a much later Cree line, so it allows for more Notch to happen early on, but it delays it later on. What you can see are these like holes, these cyst-like things that are forming, and these are actually corticomedullary cysts. And we actually know by staining that they're actually of proximal tubule origin, okay? So this is a normal nephron. If you delay Notch at a very early stage, you actually don't get proper nephron formation. And this is one of the reasons why you get the smaller kidneys and dysplastic kidneys. The other thing is that if you delay it, the inactivation, you actually get proximal tubular cysts. So these are like enlarged tubular diameters, okay? So one concept that I want to tell you is that as you inactivate more and more of the Notch signaling pathway component, you get much more of a severe disease. So here you have Notch1 and 2. Both of them are transmembrane receptors, right? So if you inactivate both of them, right off the bat, you get multiple cysts right at birth, and these kidneys are actually smaller. And the DNA binding factor, which mediates the Notch signaling, they also have a much more severe kidney disease. So if you look at this, basically what I'm showing you is this is in the wild type setting, and here you're inactivating more and more of the Notch signaling pathway, where finally out here, you're basically at sort of a zero level of Notch signaling within the kidney. And you're looking at here the percentage of mice that develop cystic kidney. So as you dial down the level of Notch, you get to 100% of the mice developing cystic kidneys. One thing to note is look at this guy right here, where you've inactivated Notch2. So a lot of mice have the same genotype, but only 23% of them develop cystic kidney disease. Okay, so what does this mean? So this is what I mean by a sensitized background, and this is maybe what is going on in the humans in terms of the level of Notch signaling. Even though the genetics is the same, you don't get the same outcome. And this is a sensitized background in the sense that if you add an additional mutation, just additionally mutate one copy of Notch1 on top of the inactivation of 2 copies of Notch2, you jump from 23% all the way to 100 percentile. So it dramatically increases the disease. So a minor mutation that normally doesn't matter is going to start mattering and increase the severity of the disease. So what I've told you is that the degree and the timing of Notch signaling matters. So as you inactivate more and more Notch, you get more and more severe kidney disease. So the inactivation of, and also the other thing I've actually told you is the inactivation of additional Notch signaling pathway genes in addition to Notch2 increases the severity of kidney disease. So here we're looking at the renal function, a very crude measure, blood urea nitrogen levels. If you inactivate Notch2 by itself, right off the bat, there is no defect right at birth. But on top of this Notch2, if you just inactivate one allele of Notch1, you start getting this severe kidney disease. Again, suggestive that there's sort of an additive function for Notch1 and 2 within the kidney. Here's again the lifespan, and I won't really go through it, but basically it's the same idea. On top of Notch2, if you inactivate Notch1, the duration during which they actually survive is reduced. Okay, so basically both Notch1 and 2 both contribute to the functioning of the kidney or the development of the kidney. This actually shows you the size. As you dial down the level of Notch, it's less and less likely that you're going to get a normal kidney. Okay, and I'll actually skip over that. So what about it in humans, right? So again, I told you there are, this is again from Dr. Spinner's lab. So you have mutations that truncate the Notch2 that result in kidney disease, and the kidney disease is sort of variable. Why is there so much variability? Is it possible that there are additional mutations in additional Notch pathway genes, or in cystic kidney disease genes that might enhance the severity. So one of the things that we've sort of gotten into, and partly it's because of the Alagille Syndrome Alliance, without which I probably would have never ventured into this, and maybe I shouldn't be. But anyway, so with the ability to do whole exome sequencing, we're actually able to look more unbiasedly at all the genetic contributions to diseases like Alagille syndrome. So one of the things we're interested in knowing is whether there are additional genetic variations in addition to Jagged Wan and 2 that might increase the severity of the disease so that it becomes early onset or a pediatric disease as opposed to an adult or late or no disease. The other thing is, just because we are interested in knowing whether additional Notch pathway components are involved, we're interested in looking at that small minority of people who don't have mutations in Jagged Wan or Notch2. So this is one of the reasons why Alyssa was talking about and why we're trying to collect a spit from some of you guys. We're really interested in people who have kidney disease, especially families that have sort of multiple people who have Alagille syndrome, and they have sort of variable kidney disease. So if this is one of you guys and you would like to participate in this, I would love to speak to you. So just to briefly tell you, so as a test, we had collected some DNA samples from some of you guys, and we were just checking out the pipeline, whether we can actually do the whole exome sequencing, and this is far from complete. This is very, very much preliminary. But one of the things here you see is in this family 3, this patient reported that they don't have a mutation in Jagged Wan or Notch2, and what we identified is sort of a rare variant within Notch1, okay? But it's far from certain whether this is actually a causative mutation right now. So it is a rare mutant in that we compared it to published genome sequencing, and it's not really there in that instance. So it's a rare Notch1 variant. We still have to we haven't even validated whether the parents who are reported to be unaffected, whether they have this or not. So, so it's not a certainty that this is actually a causative thing right now. We're also trying to test the ability of the Notch1 variant to be activated by Jagged Wan and Delta 1. One of the interesting things is we have validated this mutation by Sagner sequencing. So you can see there are sort of 2 calls at this position, a C and a T. And what that will do is that in exon, in the epidermal growth factor repeat 26, it actually changes the conserved serine. So both in the mouse, that stands for M, and H in the human, this region of the protein is highly conserved. This conserved serine actually is changed to a leucine. And if you guys were around for some of the early scientific talks, some of these serines in the EGF repeat, in fact, this particular one in EGF repeat 11 is actually modified by sugar, which is actually an important feature. So we're actually collaborating with Takuchi to figure out whether this particular serine is also modified and if it's functionally important. We are actually more interested in people with a variable kidney disease, and that's the reason why I'm showing you this family, and we're still chasing after this. We've only started working on a genetic basis within this family, but here you see that there are multiple family members with kidney disease. Some of them weren't really thought to have Alagille syndrome, but they very well, and they haven't had sequencing done, so they very well may be Alagille syndrome, mild forms of Alagille syndrome, but some with kidney disease. So the final thing that I want to tell you about, well, one thing I want to tell you about is that what kind of mutations might modify the severity of the disease, okay? And one thing is that Notch signaling deficiency is actually associated with abnormal cilia. So cilia are these antenna-like structures, all right? In the wild type, you have this tiny little cilia, and it's actually a signaling center. A lot of signaling activities happen there. And in the Notch mutants, what you can see, so here's one cell. The cilia are a lot longer, okay? And this is something that we've actually confirmed even in cell lines, where we block Notch in an interesting manner transcriptionally, and there you can see these longer cilia compared to the short cilia in the wild type setting, right? So the Notch mutants have these long cilia. So why are these cilia important? What we think is that they're probably involved in the normal tubule morphogenesis. So normal tube, structures that form tubes are likely to require the normal length of cilia in order for normal signaling to happen. So if you grow renal cells in collagen matrix, they actually form these sphere-like structures, this ball-like structure with one single lumen, all right? So this is sort of an assay that we use. So when we block Notch, they actually don't grow very well, and they actually don't form a proper 3D sphere with a single lumen. Instead, they form these small ones with multiple lumens. So there are a lot of genes that have been associated with cilia, and maybe these are good candidate cilia modifier. At least these are potentially at least modifiers of the severity of the kidney disease. At least that's one thought. So the final thing is the diet, and then really where I'm coming at from is that we've more recently inactivated Notch in the adult setting, adult kidneys, and what we see is that there's actually an active function for Notch in maintaining the cell types there. Okay, so within the collecting duct, you have this red cell called an intercalated cell that's involved in pH regulation, and these green cells express aquaporin 2, and they're involved in water regulation. The red cell actually expresses Jagged Wan and Delta 1, the ligands, and it actually signals to its neighbors to tell them to remain the water-regulating cell. So that's what you see here. So you have, in normal setting, one red cell and a lot of green cells. Now, if we actually inactivate Notch in the adult, and that's what happens is that you lose all the green cells. Their reminder, their Notch signal is gone, and so they all flip to this minority cell type fate. So what this means is that you don't have enough cells to reabsorb water, and so these mice actually have a urine concentrating defect, so they pee a lot more, they need to drink a lot more water. So we think Notch actually is actively involved in the adult state to remind cells what to be. And why is that important? It's because there may be certain instances, and there are sort of clues in the literature that certain types of diet might also cause this kind of switching, and that's kind of where I'm getting at, as the diet may be an important factor in terms of what cell types remain, because they may actually influence the level of Notch signaling, and therefore the cell type might change in accordance to the diet. And that kind of switch in cell type may not be happening normally in human patients suffering from Alagille syndrome. So anyway, so this is my concluding slide. Quite a bit of the work was started in Rafi Kopan's lab while he was at WashU and while I was a postdoc in his lab. It was there that made the 6-degree Notch2 mouse, and actually one of the first mice that I opened up that had a cyst, I think it was around the time that Dr. Spinner published that 2006 paper linking Notch to cystic kidney disease. So anyway, I'll take any questions.
Thank you, Kamesh. So